Thank you. Thank you, Luis and Bernd and the entire Dart team for the invitation to speak today. Do you guys hear me well there?
Yes, all good.
Yes? Okay. All right. So, yes, it's great to present once again at this fantastic workshop. So Chris Brauer and I have a session prepared for you about natural selection, trying to identify environmental effects on genomic variation. And, the entire planet faces an unprecedented biodiversity crisis. We have many serious issues that we have to deal with, especially related to fragmentation and deterioration of habitat. But it's great to see so many young, bright people interested in using biodiversity genomics to address these issues. And this workshop provides the ways for you to make the most of your sequences and hopefully understand a little bit more about the evolution of biodiversity and come up with management plans to protect threatened biodiversity. So what we have addressed for you, or what we have prepared for you, is a session that talks about how to collect information about adaptive diversity out of your sequences, in addition to information about neutral variation. Just before I go into the structure of our session, just a couple of minutes about our lab, which is the Molecular Ecology Lab at Flinders University. We integrate environmental and genomic data sets to address both pure and applied questions in evolution, ecology, and conservation management. Our focus is on aquatic organisms, especially aquatic vertebrates, fishes, sharks, rays, whales, and dolphins. But we also work with terrestrial animals. We have a project on koalas. We have had several collaborators and former students working on lizards, butterflies, and other creatures. The important thing about the lab is that over the years we developed a way to put together a team that learn a lot together by working together, sharing their passion for biodiversity, conservation, and evolution. And the two, in addition to Luciana Muller, who heads the cetacean projects in the lab, and I, who often had the fish projects in the lab, we have two fantastic Jedis. These are our true analytical Jedis of the lab. Chris Brauer, one of the presenters of this session this morning, and Jonathan Sandoval Castillo Juma. So these are the guys who actually managed to develop, improve, and test a lot of the codes that we use these days to try and understand natural selection in wild populations. So the session overview that we have, so I'll probably be using the first 40 minutes or so of the session with a conceptual introduction. I'll talk a little bit about adaptation, natural selection, the importance of understanding standard genomic variation. I'll then move into a brief description of what landscape genomics is, which is like a branch of the field of ecological genomics. We'll talk briefly about genotype environmental associations, and then I will finish with description of some case studies from the lab that illustrate our approaches to understand adaptive diversity in wild populations. Then we have Chris moving in. He will be doing all the hands-on coding session of a part of this session. So he will focus on genotype environment association analysis, and especially on one particular method, redundancy analysis. And he put together a lot of the text that we have available in our module in the Dart resources. Then we have a session of Q&A. And by the end of this morning, we hope that you'll be able to understand the logic of genotype environment association analysis, why it can help identify regions of the genome under natural selection. You'll be able to prepare and visualize environmental data to be used in a GEA, understand the importance of population structure. You have to account for that factor, population structure, as a confounding variable in your analysis. You'll be running a partial RDA to identify GEAs, test model significance, identify candidate adaptive loci.
Luciano?
Yes?
Sorry, I think you stopped sharing the screen.
Have I?
Yeah.
It's back now. Yeah.
It's back now.
Maybe put it in presentation mode.
Is it back? Yep.
Yes.
Yeah. Yeah.
And visualize
Beautiful.
candidate markers in genomic and ordination space. So this is what we have planned for today. But before I move further, we have a little poll just to know a little bit about your interest in understanding natural selection and adaptive diversity. Do we have the poll there, Bern? Oh, here we go. Are you considering identifying adaptive diversity, in your genomic data sets? So we have yes, no, or maybe as options for you. It's just to have a bit of a feeling about our audience. So how do we do this now, Bern? Should we have a look at the results? I cannot access them from here.
Okay, so we have.
I can't see the result. Oh, it's yes, it's 45%. No, it's 21%, and maybe it's 33%. So most people, the majority, are using it, and maybe, and see if you can convince the maybe ones.
Great.
Which are at 34% at the moment.
Okay, great. We might go back to this question by the end of the session. All right. So let's move on then. And, start with some very simple, but I believe relevant distinctions and some personal observations. Like to begin with, I think it's important to define adaptation. So we are all speaking the same language during this session. As a feature of an organism that serves a purpose in one particular environmental context. And that feature, it could be a trait, if you wish, is the result of natural selection in the past. So, for instance, we might recognize our organs, like, for instance, our vision, the eye, as an important adaptation. But it might not be relevant to understand what type of genetic variation is impacting on that adaptation if we want to, assume that the eye has an important function and we want to understand that function. And with that, I move now into the definition of natural selection, which I think it's quite important to define that as a process that is in progress. Natural selection is different than adaptation, being the gene frequencies that are changing right now in our current environmental context because of how genetically different individuals interact with their environment. Natural selection, and this is a very bold statement here, has really nothing to do with what's necessary, adequate, or not for continued survival. The analogy here is like a game of poker. The person with the best hand of cards or phenotype, in this case, will win the round. It doesn't matter if that person has a pair of twos or 4 aces. The idea here is that the best phenotype in one particular circumstance will reproduce better than others. So these distinctions are important because they relate not only to your study goals, but also to career goals. There are many folks out there that might not be interested in explaining adaptations with their data set. It's actually quite a major task to do that. And you might need separate data sets, such experiments that look at fitness trait variation in addition to genomic variation, if you want to explain how adaptation actually works. And this can be quite different than identifying the signal of selection in progress. Most of the work that I'll be talking today relates to the second situation. We are trying to use genomic data to identify the signal of selection in progress. And some folks might spend their entire career doing that without actually looking at how one particular adaptation functions in a particular environmental context. One thing that other speakers have mentioned during this week is that working separately with the genomic data sets without really digging deep into natural history can often be a major mistake. Natural history is indeed the source of our questions, of our hypotheses. And we have to understand the spatial non-random mating, in this case, population structure, and demographic history if we want to understand natural selection in wild populations. I talked about these two components first because this is something that pretty much everyone with a good sampling design and a good genomic data set can do these days. You can, in many ways, easily characterize population structure and to some extent demographic history. However, if you want to really dig deep into natural selection, you have to know the ecology of your study species. You have to go to the field and, be bitten by your study species, fall in love by it, understand the environmental context that species is found, fall in love by the habitat of that study species. Those types of things really, really matter. And for some systems out there, we know very little about their ecology and natural history. And that can be a challenge. So I just wanted to set up these distinctions and this kind of, in many ways, romantic scene to understand adaptation, which is looking at natural history as source of our hypothesis. And I think it will become more clear as I move on why this is important. But you might ask your question, especially the folks who said no to the poll. Why identifying genomic regions under selection matter? Well, first of all, we can have an idea by doing those tests of what kind of environmental variation matters to your study species.
Hello, Luciano. Can you hear me, Luciano? I think we lost your sound. Yes. Now.
Did you?
The last 30 seconds.
All right. Okay. Sorry for that. I don't know why that happened. But here we go. Please feel free to interrupt me because I don't know, if you guys can hear me or not.
Yes.
But I was basically talking about what's the importance of identifying regions of the genome under natural selection. I was mentioning the importance of characterizing adaptive units for conservation. They might be important in terms of development of specific management plans, especially if you're in a if you're in a scenario where you can predict adaptive capacity to future conditions, especially, for instance, climate change, is something I'm going to talk a little bit about it. And then you can use all this information to prioritize populations for conservation interventions, such as translocations, genetic rescue, captive breeding, to bump up lost adaptive diversity and promote population persistence. So yes, a lot of these things are possible these days, because we are in this, fantastic moment in history of biology that pretty much anyone with adequate resources out there and a good sampling design can characterize the standing genomic variation in populations. This is a central definition to understanding natural selection, because the standing genomic variation that we have in wild populations comes from a complex interplay of random events, especially natural genetic drift, and non-random events that are conditional on particular circumstances that relate to natural selection, both environmental natural selection and human pressures, because we are indeed a main component of natural selection these days. We are perhaps the strongest selective force acting on biodiversity at the moment, human influences. So it's the combination of these complex factors that will characterize levels of genomic variation in populations. Some of those, the majority of that variation, will be seen in your data set as variation that has no impact on fitness. We call this neutral variation or neutral diversity. And here you often see a signal across the genome that relates to factors impacting that diversity. For instance, 10, 20 good old microsatellites, 10 good allozyme markers might be able to characterize neutral variation population structure across the genome. However, if you want to understand genomic variation present in populations that have an impact on fitness, what we call adaptive variation or adaptive diversity, you need more markers, because that variation usually doesn't have a genome-wide signal. Sometimes it's found in only particular regions of the genome, and either controlling a phenotype or physically linked to a particular phenotype under natural selection. And to characterize that, we need more data and a different toolbox, a different analytical toolbox than the one we would use for characterizing neutral population structure. Something else that goes into this loop here is phenotypic plasticity, which is a way for populations to provide a short-term response to environmental variation. Usually this comes with variation in gene expression, for instance. So yes, let's always consider the possibility that populations are responding via phenotypic plasticity in addition to genetic adaptive diversity already fixed within the genome. And that phenotypic plasticity, if there is very strong directional selection on that, it can eventually become adaptive. So it really does matter within this context. In our workshop, we will focus on standing genomic variation, not on plasticity, both on neutral and adaptive variation. So, as I said before, you guys are in a position to do this type of work because we are living in the great era of genomics. And this plot, although quite old these days. I think it explains exactly what I mean here in terms of our ability to characterize a variation, which is along the x-axis here, that might be causing a particular variation in a particular trait or phenotype under selection. And to make a long story short here, your ability to characterize this variation under selection is dependent on the number of loci, the number of sequences, the number of markers that you have. I finished my PhD in this century. That I'm not that old, but I did work with all the tools that you can see, all the genetic markers that you can see in this, and also with allozymes, which is not even showing there. And it's quite clear these days that if you go to the top right section of this plot, and especially if you can work with full genome data sets and important resources, I'm talking here about very good quality assemblies, genome assemblies that come together with annotation of functional regions. You need a transcriptome for that, or perhaps even better, with a population data set of high quality genomes, genomes that are putting together in very good coverage. So those are the types of data sets that will give you the best likelihood of finding genomic regions under selection. This is the realm of ecological genomics. Of course, someone sitting in the bottom of this plot, working, let's say, with disease resistance in a particular mammal species, and that person might have only one marker that characterizes well variation in MHC locus, for instance. That person might be able to address their questions about selection related to disease evolution. But most of us will need lots and lots of markers. The top right part of this, it says that to acquire those data sets is expensive. But I'm not only referring here to the amount of sequences that you need. I'm mostly referring actually to the time that you need to analyze those sequences. So the genome is not that expensive anymore. The interpretome is very expensive because you need time, salary time, PhD time. All those are very valuable commodities in our field. So be prepared to put enough time to try and find the signal of selection in your data sets. Going back to this workshop, I guess most of us will be looking at, do you see my laser there, my pen?
Yes.
Yes, we see. Yeah.
So most of us here are probably working with reduced genome datasets, such as DART, GBS, DDREG. And one relevant question, that we could address is reduced genome sequencing enough to understand the signal of natural selection in wild populations? And I'll go briefly to the answer that it's still a big yes. Some folks in the past, especially the people in these particular news and views here, they wanted to break RED, DART, and other data sets, meaning that they thought that those data sets were not enough because to understand selection, because they were interrogating only a small size of the genome. But the reality is that most of us are not trying to find all adaptive loci. Our common goals most of the time are to test whether there is any evidence for adaptive differentiation, for adaptive diversity in our data set, and also to understand how this adaptive variation is distributed over space. So the reality then is that even a good quality DART data set that comes from a very well thought experimental design for your sampling can indeed be used to address questions about natural selection. You have to consider linkage disequilibrium in your data, which is actually not a big deal for folks working with threatened species that tend to have small population sizes because of bottlenecks and other factors. So those species tend to have high LD. So that's, at the end of the day, is not a big problem for them. Of course, if you work with very large populations, as some of us do, then you have to consider LD in your data and whether the number of markers you have is enough. But you have to, of course, conclude from your data in the context of the power that you have to detect effects of selection. So before I go into describing the main set of methods that we use to detect network selection and the environmental factors in genes involved in local adaptation, it's important to understand the main sources of variation that we biologists have in our hands. And they are represented by these three boxes here: the environment, the genotype, and the phenotype. So in this very simplified scheme, the black arrows here are the direction of evolution between the cause and effect. The gray arrows is how selection acts across these different levels here, across these different boxes of variation. And let's just think about, for instance, population genomics, which is most of us do. So population genomics will only interrogate information on genotypes. Quantitative genetics will interrogate information from phenotypic variation. Some of their methods, such as GWAS, quantitative trait locus mapping, they will combine the information from those two. Folks, especially plant folks, can bring their study species into the lab and come up with all these cool common garden and reciprocal transplant experiments to understand fitness traits. But most of us are not in that possibility. So then it becomes quite of an obvious choice to try to correlate the environment with the genotype. And this is the set of analytical tools that we will focus our session: the environmental association analysis of our genome-wide variation, of our standing genomic variation. There are basically two main classes of tests that we can have. I'll kind of move a bit faster now because we are half an hour into the session already. So the first main group are the outlier tests. The idea here is that in addition to genome-wide effects that we see from neutral variation, any data set, we also have locus-specific effects. And this small group of markers, they include SNPs in this case, that might co-vary with fitness traits or with environmental variation or with both factors because of natural selection. So we call these things as outlier markers. They tend to show very high FST values, and they might represent adaptive markers. There's an important thing to consider here, that these outlier tests, they tend to work okay for species that are highly connected, but not really for species that are very well structured, that species that evolve genetic differentiation over space very rapidly, which is the case of most threatened species out there. And that's because you can see that the reasons for that in this simple plot here. We have FST among loci. as a function of the probability of values of FST that you find for 3 species. And you can see that they have very different distributions of FST and very different average FSTs, from 1% to 25%. And the problem when you have highly structured populations with high FST is because there is high variation in FST among different loci. So the variance in FST values among loci increases with average FST. And that's why this third species here that has a 0.25 average FST would not be a good species to be used for outlier tests. In that case, and also for species that are highly connected, it's very important to ask the environment questions about natural selection. And that's when we try to integrate environmental and genomic data sets. That is the branch of ecological genomics known as landscape genomics. It's now a relatively well-established field. Some people believe it's about 20, 25 years old. I'll soon show you a plot of a study that was done during the 80s that can definitely be a landscape genetics study. But there are lots and lots of great reviews, including entire special issues on landscape genetics and landscape genomics. In our 3 review published a few years ago, we defined landscape genomics as the study of genome-wide neutral and adaptive variation of ecologically diverse populations sampled across heterogeneous landscapes. So in landscape genomics, most of the studies will use genotype environmental association tests in addition or as instead of outlier tests to identify natural selection. The idea here is quite simple. If you sample your populations along the environmental gradient, as we have here in this plot, and you collect environmental data from that region, you have to, of course, understand and have a good guess of what will be the variables that matter to your study species, that matter to that environmental context of selection. So in this case, we have 3 major categories of variables: elevation, precipitation, and temperature. You extract your DNA and you type this in thousands and thousands of SNPs. The idea here is that if one of those SNPs, such as the one here in the bottom of this Manhattan plot, if this SNP is under selection, the frequency of that SNP will vary as a function of how that particular environmental variable is found over space. It's a very simple idea that strong divergent selection between different geographic locations might result in local adaptation because of differences in average phenotypes. And this will create differences in allelic frequencies. And because divergent selection is very strong, those allelic frequency differences will be maintained even in the face of strong gene flow. So basically, to repeat what I just said before, those GEA tests, they identify adaptive SNPs or candidate adaptive SNPs by testing for direct associations between how the frequencies of those SNPs vary across environmental, spatial environmental variation. Some folks might be in a fantastic position to basically conclude that their results are indeed indicative of natural selection. And here we go, a study that generated data during the 70s. This is allozyme variation for Drosophila. We see that there is one of the 2 alleles in this enzyme, which is the alcohol dehydrogenase, that is found as a client in Australia. Let's look at Australia, the map on the left here. Basically, you have two alleles that are maintained by selection, because in this case, the variation in one of those alleles is correlated with variation in maximum rainfall across the Australian continent. And why you can infer natural selection with so much confidence in this system? Because that very same species in another continent is showing exactly the same pattern. We have the same allele showing a clinal association with maximum rainfall in the North American continent. So you got two Darwinian laboratories here to test selection. You have very good samples, and only one marker associated with environmental variation here might be telling you a very important story. There are problems in any of these approaches, even when you have repeated Darwinian labs, as I mentioned here, because many populations are not independent. This is something to have in mind. Let's look at this other study here. This is a huge global data set for our own species, humans. And we have the frequency of one allele, again, here, plotted over the distance of that allele from the equator, of that allele is sampled from the equator. That allele is known to give resistance, not to give resistance, that allele is known to being able to reduce the amount of sodium in our cells. So when that allele is found in high frequency, folks have less problem with hypertension. And that is the case of most of these populations here on the top right corner of this plot. It's giving a selective advantage to Europeans, for instance, because of high frequency of that allele, but it's giving problems to African populations that are closer to the equator. But although we have 52 populations sampled for this study, we know that geographic space and ancestry will create problems of independence. Many of these population samples here are not independent from each other. They have shared ancestry. So you have a smaller data set than originally thought because of this issue. How do you address these issues, such as population structure, isolation by distance, population history? There are many types of GEA tests that can account for the influence of these variables. And one important test is called the RDA, or redundancy analysis. And that will be the test that Chris will give a hands-on session for you very soon. So RDA, they model linear combinations of your environmental predictors, the variables that you think that matter. And those models might explain the linear combinations of your response variables, the SNPs in this case. And you can use allelic frequencies, and you can use individuals. You can identify then SNPs that co-vary with the environment and that are associated with these multiple environmental variables that you add into the analysis. It's considered by many as a real Swiss Army knife for landscape genomics because it's very flexible, flexible tool. Some studies have shown that RDA provides the best balance between low false positives, which is a major problem in our field because we have so many markers, right? And there is, of course, a good expectation that many of those markers will just be giving you false positives in your tests of selection. But it also gives a good value, a good balance between high true positive rates. RDA seems to be robust across a range of demographic and spatial scenarios, sampling designs, and different types of selection, including polygenic selection, which is the type of adaptation that we tend to see in many data sets across environmental gradients, the type of response of the genome for environmental variation because of climate. The idea here is that you don't have very major changes in the allelic frequencies. You have minor changes in many different alleles across several regions of the genome, because it's the combination of those very minor frequency changes that are actually controlling these very complex phenotypes that are responding to environmental variation. And RDA seems to be to have good power to detect polygenic selection. So I had several very mini cases, very small, quick runs of some of the work we've done about natural selection. I might have to skip a couple of those for the sake of time. But the idea here of describing these studies is because several of those studies, they actually use ways to validate their inference of selection that were done using GEA tests. It's quite difficult for many of us to bring our study species into the lab and validate our inference of selection doing experiments, so for instance, fitness tests, experiments of adaptation to climate change. Those things are very difficult and impossible in many cases. So the studies that I have here, some of them have validated their GEA and RDA results based on using other ways, some clever ways, I believe. So let me just quickly show you one example that relates to what often happens in the sea, where you have species with very large population size, where both sperm and eggs are released in the water column. So this is called broadcast spawning. It's a very common mode of reproduction in the sea. Not only population size are big, but there is also high connectivity in these systems, very high gene flow. And of course, we should be able to capture the signal of selection here, because natural selection, the efficiency of natural selection, scales up with population size. So things like corals, fisheries resources, they have huge population size. And we should be able to then detect environmental heterogeneity impacts on the genome despite high connectivity. I have two case studies about these types of system. I will skip the first one and just jump to the second one. And I'll only briefly talk about this work because that is the paper that Chris is using. That's the data set that you guys will be running your hands-on session very soon. This study was published 2 days ago, so it's quite fresh, and I think it's a very elegant study done on Snapper, which is arguably the most important socioeconomic marine resource in the bottom half of this continent. Now, many folks rely on snapper, not just for commercial and recreational fisheries, but also for a large chunk of Australians who live along the coast. They use snapper, and they rely on services provided by snapper, such as real estate, charters, and local activities along the coast. But snapper populations are in decline. WA and South Australia, both Western Australia and South Australia, have major bans on fisheries of snapper, both commercial and recreational. And this study tackled how connectivity in snapper can be disassociated from the neutral demographic perspective to the adaptive connectivity. And it focuses on a very heterogeneous environment, especially here represented by the two gulfs in South Australia. So for instance, let's focus on this one for the sake of time. The one that I'm showing here is the major gulf that we have in Southern Australia, Spencer Gulf. There is a huge gradient in salinity here, because there is high evaporation in this Gulf, very little freshwater input, if any, during summer, and therefore salinities can be quite high, around 40 or higher, as opposed to the 33, 35 salinity that you have in the open ocean. Temperature also varies quite a lot. This plot here is the top is the mean temperature over the year. So you can see that both Gulfs are much colder than areas of the adjacent coast. So what Chris did here is to run RDA tests, and I'm not showing those because that's the hands-on session, and use the adaptive data set to estimate migration rate across this entire coastal region. So the adaptive data set is here on the D plot of this migration network. Let's just look at the northern Spencer Gulf again, the population in orange at the top. Migration rate now it's about 0.5 between that area and other areas of the coast. But if you use the entire neutral data set, migration rates are much higher, some of them close to 100%. And we have a relatively good trust in our adaptive data set because Chris managed to predict the effects of gene function on these adaptive SNPs. And they are predicted to have quite a lot of those 800 or so adaptive SNPs are predicted to have moderate or even high function. So what we are seeing here is evidence of how environmental gradients decouple demographic and adaptive connectivity. This is extremely important for fisheries because this area here, the northern part of the Gulf, contains a very important breeding ground, a nursery area for snapper, the most important one along the South Australian coast. So moving on to another species found in the same environmental gradient, bottlenose dolphins, we have now the importance in this study of understanding sociality, how related animals tend to hang around in the same type of habitat. Basically, we also did the seascape genomics analysis here of dolphins found in the Gulfs and outside the Gulfs, but the sampling was only done after about 15 years of field work that characterize patterns of residency of these animals. So in fact, dolphins that I'm now showing here in the bottom plot that are found in the Spencer Gulf, the northern part of the Gulf, they tend not to leave that area, whereas dolphins that are found elsewhere, they are much more mobile. So basically that's that type of field-based information on social mechanisms that impact on residency patterns were used for the sampling design of our seascape genomics analysis. And this is the RDA plot. I'm not going to explain this now, but just focus on these guys here at the top. So these dolphins in this tone of blue at the top are the dolphins from northern Spencer Gulf that are found in a hypersaline environment. And we are arguing that they are locally adapted to that environment. We found the same, some of the same candidate adaptive SNPs in these South Australian dolphins in an analysis that we did using dolphin populations from around the world. We have now populations from different oceans here that are also found across environmental gradients. And guess what? The same gene regions appear to be under selection in these other dolphin populations from other parts of the world. Some are even different species of bottlenose dolphins. So that's another way to try to validate your findings of seascape genomics or landscape genomics. I'll jump now into my very last case study because I'm running out of time. So everything that I mentioned to you so far is looking at recent influences of environmental variation, recent footprints of selection. So we're looking at contemporary patterns of selection, adaptive diversity these days. What we can also do with landscape genomics is to project this this understanding of current patterns of adaptation into future climates, future environmental scenarios. And that's the field of landscape genomics that is growing in popularity. It's also very well, of course, receiving quite a lot of criticism, but we can talk this later on during the Q&A. And this field is known as genomic offset or genomic vulnerability. And is usually done in the context of climate change, future climate. So what folks can do here is to integrate your GEA information, let's say that you obtain using redundancy analysis, for instance, from your wild populations, with information from niche distribution models, both in the present and future. So that gives you an idea about your, for instance, climatic envelope of the species today. But also will give you projections of how the climate or the environment will change under different climatic scenarios, right? Or let's say business as usual, or a particular increase in temperature. And we have all those scenarios and the data sets publicly available for you to integrate that information into your study, especially for terrestrial systems, but also for coastal marine systems. Then that will give you an idea about whether your populations that are currently adapted to a particular environmental circumstances might need more or less evolution to maintain those present patterns of adaptation in future climates. And that is what we know as genomic offset or genomic vulnerability. If your analysis show that some particular populations have very high genomic vulnerability, meaning that they need a lot of evolution to maintain patterns of adaptation in the future, they are those populations that are more vulnerable to climate change and might need immediate attention in terms of management practices. Our case study about genomic offset was also primarily done by Chris on tropical rainbow fish found in the Atherton tablelands in Queensland. So here we have perhaps the only upland tropical rainforest habitat in the country. And up there you have a few species of small and threatened rainbow fish, which are being now put into contact with a warm-adapted and very abundant rainbow fish that is usually mostly found in lowland areas of the wet tropics. So when that lowland rainbow fish is spatially in contact with these different species of threatened upland rainbow fish, where they meet, they tend to form hybrid zones. And what we found is that in the hybrid populations, we see a bump in diversity that is related to climate, that is associated to climate. We believe this is warm adaptive diversity that was brought from the lowland species into the upland habitat and was introgressed into these populations through hybridization. So what Chris did in this work then was to estimate genomic offset for the pure upland populations and the hybrid upland populations. And the results are summarized in this plot here on the right. So here on the y-axis, we have the amount of evolution that each population needs to adapt to a future climate. And on the x-axis, we have the ancestry of the warm adapted species in these local upland populations. So as you can see, populations that are pure upland, no hybridization, they are in circles here, they need more evolution to adapt to a projected climate than populations that have hybridized with the warm-adapted lowland species. So we are concluding here that natural hybridization has reduced vulnerability to climate change in this system. And we are now using ecophysiological experiments of thermal tolerance in a new study to try to validate these conclusions from genomic offset. I will stop here. So now we have Chris moving into the hands-on section. Just before I do then, I have two shameless plugs here for you. I'm one of the editors of a joint special issue of Molecular Ecology and Evolutionary Applications on understanding and adapting to climate change. So if you have a very cool paper addressing this topic, get in contact with me or any other editor listed in these two journals. We have a submission deadline of those papers of 30th of June this year. And I'm also looking for 2 PhD students to join the lab to work on adaptive introgression and rapid evolution to climate change. That Rainbow Fish project I just mentioned to you, and the scholarships are available both for domestic and international students. I will stop now and pass on to Chris to talk about RDA. Thank you very much.
Very quickly, there are a couple of questions. When do you want to answer them?
There's quite a few. Would it be okay? What do you think, Chris? Should we go into this after your session? That would be my preference at this stage. I cannot hear you, Chris.
Yeah. No, I can't.
Okay. So let's address the questions after Chris finishes, because some of the answers might be in there. I still can't hear you, Chris.
Okay, let's start with some questions while Chris is ready. So maybe we can go and start with the first one.
Is that the one about scale?
Yes.
Yeah. Well, it's context dependent. Yes, it would be very hard to guess without knowing much about not only the species, reproductive mode, dispersal potential, life history, but especially about the environmental context of selection. So yeah, it is indeed context dependent.
Does it turn green when you say something?
We can go to the second one.
Okay.
Yes, we can go to the second one. So I can read it for you. I love the talk. Thank you. I was wondering how gene function was predicted in the snapper example.
This was through SnapF. Chris can give you more detail because he did that work. I just know how it was done. Chris, do you have a mic now?
Yes, I believe so. Can you hear me?
Yes, perfect.
Okay. In a second, when I share my screen, I actually saw that question and I'm getting some weird feedback now.
So the method is known as SnapF. We also have a very good genome for Snapper that is annotated with transcriptomic data. All of that stuff matters. Are you back, Chris? No, you are not. And the details about the analysis are on the paper. The paper was published this week as open access. Can we go to the third question, maybe?
Yeah, this is, thank you for your talk, Luciano. Your point on phenotypic plasticity was very interesting. Could you explain more about how it becomes adaptive and how we may see it in the genome?
Wow. Thanks. Very, very difficult questions to address. Basically, phenotypic plasticity can basically get trapped in this environmental context for divergent selection, meaning that the same response that provides a buffer to environmental variation might be required over and over. You could think this as maybe a seasonal variation, could be a heat wave. And if the same set of responses is produced in every time that this environmental context takes place, plasticity might be selected at the gene level. Usually this can happen through gene expression variation, regulation of some important metabolic genes. Some folks believe it can also happen through epigenetics. How we may see that in the genome, our work, we were basically looking at gene expression in an experimental setup where we were looking at future climates and seeing how populations from different climatic regions responded to the same experiment. When we identified the genes that were responding to the experiment, we asked the question of, do we have candidate adaptive variation in our landscape genomics work, which only use these SNPs, that are sitting within these same important genes that are responding to future climates? And we found, yes, that there were some genes in common. So meaning you compare the results from an experimental analysis, in this case, gene expression in future climates, with your candidate adaptive SNPs from your landscape genomics. It requires a lot of work, but that's how we did it. And I'm sure there are better ways to do it, but it depends on your study system.
Okay.
Okay. And now, is that okay?
Now, Gene,
okay.
Maybe we should leave time for Chris to run this, and then we go back to the questions, because I think it's very important for people to know how to run GEAs.
Excellent.
Okay, so I think I can share my screen. I think that's working. See that? Cool. I just had a quick get this up. I saw the question about SNPF before, so that's the software, the website for the software that we use to try to characterize different variants as whether they have an impact on, or likely to have an impact on, gene function, and it can do a whole heap of other types of analysis like that too. So it's definitely worth having a look at that. Just get it back now. Okay, so I think Luciano did a really good job of introducing the session here. Some of the information I think he already went through, but yeah, basically I've got some code here that you guys can run with either the example data, which will be already loaded in the session, or you can copy and paste the code into your own local session and use your own data as well. So basically, we're just going to be running a genotype environment association analysis, and like Luciano said, focusing on redundancy analysis. And so again, a lot of this was covered really well by Luciano. And so I think the main part in this section is just thinking about what it is that you're doing in your study, and why you might want to run a genotype environment analysis. So it's really important right at the beginning to think about what are the questions that you're actually trying to answer. Are you interested in understanding which genes are important for adaptation to some environmental variable, whether it might be salinity or temperature or whatever it is? Or are you more interested in trying to understand, well, what are the main aspects of the environment that might be imposing selection on your particular population? And so you might have more of a focus on trying to understand how the environment varies relative to your species and your population. Then that might be more important for your question than actually getting to try to answer which bits of the genome are important for that. So it's really important to have a clear idea about what it is that you want to try and achieve by running the analysis. And in some ways that might depend on who you're doing it for too. So you might be working on a project that's providing information to a natural resource management agency, a fisheries agency, or other conservation agencies, and the information that they need might dictate the way that you go about running the analysis. So that's really, I think, the key point here is understand what it is you want to do before you start coding and running analysis and trying to interpret it. And so Luciano's already sort of given a really good introduction about what genotype environment association analysis is. And so I think we might skip over that a little bit in the interest of time and getting to running some actual code. But just quickly, why redundancy analysis though? There's certainly a whole host of other approaches that people have and do continue to use, and they have different sort of advantages and disadvantages, I guess. And so redundancy analysis basically is what's a multivariate constrained ordination. So in some ways, if people were watching Arthur's talk yesterday, he gave a really good introduction to PCA and sort of a little bit into the mechanics of that. And RDA is not so dissimilar to that. In fact, a lot of the I quite liked his 7 deadly sins setup. That was sort of the structure of his talk, and all of those I think equally apply to RDA as they do to other ordination methods such as PCA or PCOA. So that was a really nice introduction to have yesterday before I have to talk about redundancy analysis here. And so really I guess the strengths of this approach is that you're able to model sort of more complex environments in the one analysis. A lot of other genotype association analysis approaches you can only model sort of one variable at a time. So you'll need to run a separate one for temperature, for salinity, for whatever else you might be measuring in the environment, and it's kind of assuming that those are independent, which is, of course, very rarely, if ever, the case. And so I think being able to model multiple aspects of the environment in the one analysis is a real strength of this approach. The other one is that it has a really natural sort of useful ability or function to be able to partition the variants that you find in your SNP data, in your genetic information, not only to your model of the environment that you use, your environmental variables, but also other things that you might want to try and, I guess, sort of partial out of the analysis, or try and not include in your analysis. And the big one for this sort of stuff is the neutral population structure. So if you have a population that might be subdivided, and different parts of the range of the species might be connected by varying amounts of migration, connectivity, then you're going to get patterns in your genetic data that might mimic sort of associations with the environment, because the environment varies over space in the same way that structured populations might vary. And so if you're not thinking about the neutral structure within your data, then you can quite easily sort of, or that information can confound your assessment of local adaptation. And so what we're going to be running today is called a partial RDA, where we actually incorporate information about the neutral population structure, and try and account for that before we start looking for the associations between allele frequencies and the environment. You can probably skip over the rest of that there. So just a couple of the other sort of more common approaches, like generalized linear models, LFMM, Bayesian, all of those are usually run in a way that you can only run one environmental variable at a time. They can have really good power and are useful in some settings, and they're usually fairly easy to interpret. So they're fairly well understood methods of assessing significance and that sort of thing. It's not always easy, depending on the methods, though, to control for that neutral population structure, like I mentioned. And just as I said before, it's assuming that your environmental variables that you have are kind of acting independently. It's not straightforward to run each one separately and then try to merge your results together and interpret everything as one. So I really like the ordination methods like RDA, and there's a couple of other ones, CCA and distance-based RDA, which are all kind of similar. I'm not going to go into the details of those here. And just at the bottom here, there's a really useful simulation paper here. I think if you're interested in comparing the different methods and where they may or may not be particularly suitable, so I suggest people to read that paper there. Okay, and again, Luciano gave a really nice introduction to the study system we're going to be looking at. Just a little bit of a rundown on what the actual data is. So it's obviously snapper along the southern Australian coast, mainly sort of in the far west of South Australia, over on the left of the map here, and then the most eastern point is at Lakes Entrance here, to the east of Melbourne. And I guess the main biological or ecological features of this to think about are the gulfs and embayments. So we've got Port Phillip Bay and Westport Bay in Melbourne, and then Gulf St Vincent and Spencer Gulf in South Australia. The Spencer Gulf and Port Phillip Bay areas are really important spawning grounds and nursery habitats for the species. And there's been a lot of work done already for many decades on snapper, trying to understand how these spawning grounds actually feed the larger population at a regional level, and where the fish come from, and how that might vary over time. There's some really strong oceanographic currents that run most of the time west to east in this particular part of the country, but though the strength of those currents can vary quite a lot seasonally, and the distance offshore can matter as well. So sort of close to inshore, you actually get a little bit of a counter current in some seasons that that might push larvae in the opposite direction. So it's really quite complex trying to understand how the environment kind of modulates the broader population structure in this species. And that's before you start thinking about anything to do with local adaptation. And so in the paper, we had about 450 individual samples from the 16 kind of different locations that are on the map here. They then color coded into sort of 10 broadly sort of regional populations. So some of the individual sampling sites might have only had a handful of samples. So where there's sites that are really close to each other, for some of the analysis, those samples are combined into one site, and that may or may not be something you can do with your particular study system. Snapper, at least in terms of broad population structure, they're large populations, really well connected. So if you're sampling a fish from nearby sort of fairly proximate locations, you're unlikely to be running into trouble by sort of sampling more than one population kind of thing. So you're not going to find a Walland effect or any other issues with combining sampling sites that maybe if you were looking at a freshwater fish in the Murray-Darling Basin from perhaps two streams that might be fairly close to each other, they can be very, very different populations. So you really need to think about your particular species when you're either designing your sampling regime, or in this case, when your samples are provided by fisheries or something like that. You don't have a lot of choice in where your samples come from. Ideally, you could sample, like Craig was talking about yesterday, you'd have a randomized sampling design. In most cases, you don't have that. So you've got to try to think about the best way to approach any analysis given the constraints of your particular study. The full data set, we ended up after sort of filtering this was DD-RAD data. We had about 14,500 SNPs. The tutorial data set here, I think, is about 2,000 of those. I just made a little subset to make everything run really quickly. And then the environmental data was extracted from a data set called BioOracle, which we'll talk about again in a little bit. Session overview, we're just going to talk a little bit first up about the environmental data and potential sources of that and what you might need to consider in terms of scale and ways that you can actually visualize the data and maybe start to think about which variables might be important for your system. Just how to format your genomic data then in a way that you can run the analysis. A little bit, I'm not going to talk about filtering too much, imputation a little bit, and then we're going to run the analysis, and then we're going to go through a couple of a little bit more code after that for what you can do once you've run your analysis to try and get a little bit more information out of your data and out of your results. So we just, we'll get right into it. So the first page here in the setup, this is all just an example of the data that we're looking at, the packages that are used in the tutorial. If you're running this on your own local session, you might need to copy and paste this and run it like that, but you don't need to do this for the tutorial. It should all run within the session. And so there's just a little bit of a explanation of the data we're using. So we're using a population allele frequency data set to run the analysis. So for those 16 sampling sites, And as I said, for the tutorial, about 2,000 SNPs. The environmental data is downloaded from via Oracle, and then we've got both the raw data and then some scaled data then to be able to put everything on the same scale, so you can compare vastly different environmental variables that might something. The range of salinity is very different to the range of temperature, for instance, and so transforming them with Z-scores is a way to try and make them more directly comparable. As an omega matrix is essentially the allele frequency covariance matrix that we're going to use as our model of neutral population structure. This particular one was generated with the software Baypass. There's other ways to do that, of course. And then there's just some information about the samples and the geographic coordinates. What else? And then there's a little bit, the last part of the session, we're going to be generating a Manhattan plot to look at the location on the genome of our candidate loci. And for that, we've got some information from a VED file, which is ultimately, it came from a VCF file about the genomic position of our SNP markers. But we'll get into the thing. So the first decision that you need to make when you decide, after you've decided that you're going to run a GA analysis, is what environmental data should I use? And you really got to just start with the biology or the ecology of your species. So you need to think about what's important. You might already know that. There might be a lot of ecological work that's already been done. For other species, maybe you don't. And that might dictate the types of variables that you might be considering. If your particular study system is on a really small spatial scale and it's really well studied, and there might be particular environmental challenges that you're looking at, maybe it's pollution, maybe there's something really specific, you're going to find data from a different place that you might be if you're looking at a broad continental scale where you're probably going to be restricted to climatic data, maybe a few other derived things based on climate, but at that scale, that kind of dictates where you might look for your environmental data. And if there's nothing available, if you're starting completely from scratch, maybe look at co-distributed species. If there's anything in the literature that you can find that some part of the environment that affects those species, if they're similar, but you've really got to come to it with some idea about what you think is important. And then, looking at the data that is then available for your region and at the scale that your sampling design is. So just the example I had there, if you have a really small spatial scale that your study is situated within, then broad scale climate data that has a resolution of sort of 1 degree isn't going to be informative at all, because it's not you've got to match the scale to your samples. I'll skip over this, but I'll put some links here for some sort of the most common sources of environmental data that people use. Some of these are sort of global. There's a few down the bottom that are more sort of Australian oriented, and there's definitely more than the list I put here, but it's a good place to start looking to get you going. The next bit of code here, I didn't make it interactive because the spatial data is quite large in the raw, in its raw form that you can download. But this is essentially the code that I used to download the data for the paper. So you can copy and and paste this and go through and figure it out for your own data. And there's just a couple of different R packages here, so SGM predictors and geodata that are specifically for downloading particular environmental data sets. But having done that, you've downloaded your environmental data. The first thing you want to do is try and get a little bit of a feel for, well, which environmental variables actually vary across my study system. Because if you choose something that has no variation across where your samples are from, then you're not going to find any signal with that variable, even if it might be important for the species. If there's no variation among your particular samples, then there's nothing to correlate. You're not going to find a signal. So the first thing you need to find is which variables actually have a signal across your study system. So if we run the first one, the code box there, we can just look at our data frame of environmental data, and then you get some summaries as well. So the variables we're looking at here are the minimum temperature, calcite concentration, pH, salinity, and primary productivity, the minimum averaged across annual primary productivity. And so that's sort of the first look you'll get at your data. If you're like me, it's kind of like to see maps and pictures and things. And so the other thing that's nice to do, if you run the next one, hopefully, there we go, is to then take those layers. I've just got a couple here, so minimum sea surface temperature and mean sea surface salinity, and map those across your study system. So we can see that temperature, we've got a pretty strong gradient of variation across the whole study system. If we look at the much cooler minimum temperatures in Port Phillip Bay, and then the two Gulfs do have cooler minimum temperatures than the outside coastal waters. And then salinity, you can see the main gradient here. It's a little bit hard to see on this scale. There is variation along the coast, but the main gradient is obviously in the Gulf, so particularly Spencer Gulf here. Okay, what were we up to? The next bit, I might skip over this a little bit. Basically, any type of regression analysis, if you're including predictors in your model that are correlated with each other, they increase the total variance in your model, and which is basically, it just adds noise to your model, and it makes your model less stable, I guess, the results. So it's something that you need to think about, because a lot of climatic variables will be correlated with each other. And so thinking about if you might end up with a set of 10 or 12 variables that might make your shortlist. You want to try and remove any or one of any pairs that might be highly correlated together or vary sort of together. So there's a couple of different ways to do that. One is just to look at pairwise correlation. And so the code here, which again, it's not really, we've already done this step for the data in the session, but you can play with this. Basically, there's two steps here. One is to look at just keeping one variable out of any pairs that are greater than a 0.7 correlation coefficient. I think this was Pearson correlation from off the top of my head. And then using another type of analysis called variance inflation factor. So basically what that does is it takes your group of environmental variables and it builds a model based on all of them except for one, and then it measures how much the variance increases in the whole model when you add that variable back in, and it does it iteratively for each variable, and it figures out which of your variables are adding the most noise to your model, and it's a way of prioritizing which ones you should keep in your final model. But you can play with that with your own data. And so I spoke briefly before about standardizing your variables, and in this case with z-scores. So we can run just a quick little bit of code here, and this is taking that data frame of raw environmental data for each site. It's converting them to z-scores and then just plotting a heat map here, so you can see your sampling sites down on the y-axis here, and then each one of the variables across the x-axis, and you can have a look at how each variable tracks across all of your sampling sites. So pretty easily here we can see that in the map. If you remember, the Spencer Gulf had that really strong gradient of salinity, and northern Spencer Gulf, so right at the top of the Gulf here, that's the highest salinity there, and then the rest, there's not as much variation, but it's much lower. And then southern Spencer Gulf a little bit is high there. So you can see all of your variables on the same scale. And there's just a couple of exercises there just to get you thinking about the data, and then you can change the code with colors and however you want to format your heat map. This is just an example. And then you can save that as some code there about saving it to a PDF file as well, so you can remove it from your R session and have it later on as a figure. So we'll keep moving on to our genomic data now. And as I said, we're going to be using population allele frequencies. In this, you can use genotype counts at an individual level. It depends on your system, and right at the start, one of those thinking about which questions you're trying to answer. But in either case, and as Arthur said yesterday, ordination analyses don't cope well or at all with missing data. You need a full data matrix. And so with any genomic data, you're probably not going to have that to begin with. You're going to have missing data across your samples. And so you need to think about what is the best way for your data to account for that. And so you could just remove loci that have missing data. You might not end up with many remaining, depending on the type of data. You can just drop individuals that have missing data. In most cases, it's going to be all of them, or most of your samples will have some level of missing data. And so the answer then is to somehow fill in the blanks in the most logical way that you can think of. So again, depending on your study system and how much structure there is, you might just use the most common genotype across all of your samples. That might be okay, that might be good enough. That has, in effect, that's a fairly conservative way to do it as well, because it's making your samples all appear a little bit more similar to each other. So if you've got a structured or if your samples include population structure and you're using the most common genotype across all of your samples, you're kind of ignoring that structure and making everything a little bit more similar. If it's highly structured, you might say, oh, okay, we're going to use the most common genotype within that particular subpopulation, which is, I think, often a pretty nice way to do it. And a variation on that, which we used here, is to run a structure or admixture type analysis. In this case, it was SNMF, which I should have written down here what that stands for. The name escapes me. No, it doesn't matter. It's like admixture or structure. Essentially, then within each of the clusters, it's giving the most common allele for the population that your particular individual is assigned to. And so Arthur went through this. I think dartR has a method for imputing genotypes these days. This is one in a Millfield package, which is just a kind of a collection of mostly useful scripts that I've put together in our lab. And it just basically it'll take your Genotype object, it'll run an admixture analysis, and then it will impute the missing data based on the results of that. But this has already been done for today, and if we look at our little frequency matrix here, basically down the left-hand side, each row is a sample, and then like a sampling site, and then for each SNP you have a minor allele frequency. And then, oh yeah, that's just the range. That's just a test to make sure that your allele frequencies are actually bounded between 0 and 1, and you haven't done something weird with your data preparation. So it's always nice to have little safety checks like that to make sure that your data actually makes sense. The next decision you need to make is, do I need to account for population structure? And if so, how do I do that? Because there's many different ways. If you've got a single population or something that's highly or close to panmictic, maybe not. In the simulation study that I mentioned before, they make a fairly strong case that for even up to moderate levels of structure, RDA is fairly robust without including any control for that. But in my mind, it just makes sense that if it's something we should be doing, and I think perhaps that just stems from a lot of my experiences to do with freshwater fish, where there's a lot of structure anyway. So it's something that I've always thought about, and I think in most cases probably makes sense. You can just use simple XY coordinates or some derivative of that, polynomial or something, as a basic model of the geography of your sampling sites. There's other more complex approaches there, principal coordinate-based ones, Moran's eigenvector maps. People use FST-based sort of as a distance matrice. Admixture Q values I've seen. I've just put some notes down on the side there for, I don't know, generally what I've found with my data anyway. Some of those are more conservative than others. Some of them, I think, run the risk of throwing away a lot of signal than others. The thing that I like when you're doing things at the population allele frequency level, I've found the allele frequency covariance matrix to be a pretty good approach that does a good job of capturing the broad spatial structure in your data without necessarily throwing away too much of the signal you're after. And that's the approach that we're using today. Okay, so we have our environmental data, we have our allele frequency data. It's time to run the model. And this bit of the code here is just an example of, why did I put that in there separately? I'm not sure why that bit's at the top, because it's the same as the bit that we're going to run. So you can go ahead and run that model there. And so the basic model is essentially your genomic data, and your environmental variables, and then conditioning or controlling for your model of population structure, in this case the omega matrix, or the allele frequency covariance matrix. And so when you run that, hopefully it should run fairly quickly because it's just a subset of the data. You get a table like this, and basically, in this case, or they even put it there, the term they use for the variance in the model is inertia. In this case, this is using the RDA function from the vegan R package. And so we've got a few different lines here. So the total, so this is the total amount of variance in the model. Proportion should be 1 because it's all of it. And then we've got a couple of different things. So the conditional inertia or variance. So this is the variance in the model that you can attribute to the population structure, or in our case, the omega matrix. So this is the neutral population structure, how much that omega allele frequency covariance matrix how much of the variance in your genomic data that explains. The constrained proportion, that's the bit that we're interested in. So this is the proportion of your allele frequency data that is explained by your environmental variables in the model. And that's after partialing out the variance associated with the neutral population structure. And then you have a whole pile of variation that isn't captured by either of those two things in your model. So this is the remaining variance that isn't explained by any of your other environmental variables or your neutral population structure. And then it gives you some other information about the amount of variance that's explained on each one of those constrained axes, so the part of the model that's related to the environment. And then the PC axes here are the amount of variance associated with the remaining variation. And so we can summarize all of that. We run the next set of code. And so we use an adjusted R² value for that. And this is basically an effect size, I guess, estimation for the model. And so we go up here. In the raw information on our model, the raw results, our constrained proportion was about 50% there, 0.51. Now that's of the total variation. Now if we come and look at this, this is saying it's about 0.3, and that has to do with the way that R² is calculated, particularly adjusted R². But essentially, those two numbers are giving you the same information. The adjusted R² is just a, I don't know how I would describe it. It's, I guess, a more accurate representation of the actual amount of variation explained by your environmental variables. And again, then you can look at the amount of variance on each one of your axes. We can plot that. We can see that in our model, the first axis explains far and away the most variation in the model, and then each successive axis there is explaining less, which is a pretty standard thing if you're familiar with PCA or any of those type of ordination analyses. The model significance, you can test that at the significance of the entire model, and then you can test the marginal significance of each of your predictors. So it's a way of trying to understand, or in isolation, which of those environmental variables contributes sort of independent information to that model. And I've just got a little box there about interpreting that, because especially the marginal tests by predictor, it's not necessarily all that intuitive. So you might find that only one or two of your predictors might be, you'll get a p-value for this, and only one or two of them might be significant. It doesn't mean that the non-significant variables aren't important in your model. It just means that they are providing less of an independent signal to the model than the ones that are significant. Your overall test, the one at the top, is really the key validity check of the model to say whether, as a group, your environmental variables can explain variation in your allele frequencies. So it's something it's not necessarily straightforward to get your head around, but definitely the overall model test is the one you should be focusing on. And that's all good to look at tables full of numbers, but then we can always get a little bit more information, I think, by actually visualizing stuff. So the typical way that people will present the results of an RDA is what's called a biplot, where you can include the information about the SNPs in there. In that case, it's a triplot. I usually don't find those particularly useful, so I usually keep it to our sites and our environmental variables. And so here we've got a plot of just the first two axes, and the percentages here. You can make an argument that these percentages aren't actually very informative. If you leave them off, reviewers will complain. So I tend to include them anyway. But basically, in RDA1 here, the 70%, that is 70% of this constrained proportion. So it's not of the whole model. It's 70% of the 50% that's explained by your environmental variables. And it's, again, it's not necessarily that informative, but it's something that reviewers always ask for if you don't put it on there. And so a couple of things here. The two sites up here are these northern Spencer Gulf sites, and they're right at the high end of our salinity gradient in this view of the model. And then we've got the main axis, RDA1, splits the eastern sites that are more centered around Victoria, and the sites here on the left are the South Australian sites, so that's the major separation. And then, yeah, salinity and temperature are kind of splitting that second axis. And yeah, I've kind of answered some of those questions already. It's just some things to think about in the discussion there. And just a little, I guess, I don't know, tip, or I think Arthur did a good job of explaining the way that looking at just one view, so RDA1, RDA2, the first two axes, doesn't tell you the whole story about how your data, or the shape of the data in this model. And so one of the, if we run the next one here, that not only applies to the data points of your sampling locations in this case, but also the way that our vectors, our environmental variables, appear on the plot. So the length of these vectors represents how important it is to this view, so your first two axes. If you display different axes of your model, the length of those will change, because environmental variables, different variables will be more important along different axes of the model here. And so when we've run here we've got still the first axis on the X, but now we've got the third axis as our Y here. And you can see, for instance, pH and primary productivity were perhaps less important than calcite here, whereas these variables are a bit more important when you're considering the third axis here. So to really get a proper feel for that, I'm hoping this is going to work, you can generate a 3D view based on the three axes together. Burn.
There's my error. Doesn't it run?
Okay, it doesn't look like we're going to get that anyway. It's
That's a shame. Can you run it again? Just for fun. Restart and run. Yeah.
Did it work for you?
I'm there at the moment.
I think we might move on. But anyway, the code there, if you can trust me at all, it will generate a 3D version of that, and you'll be able to rotate that around and have a better idea about how those environmental variables actually relate to more than two axes at a time. Okay, so we've run our model, and now it's about interpreting it and coming back to what is this telling us about the biology. So one thing we might be interested in, well, which parts of the genome, which SNPs are the ones that that our models show are important and are related to our environmental variables. And I don't have time to go through this as much as I was going to, but there's a really nice function called rAdapt that is a good way of identifying candidate loci from an RDA model that incorporates information from all of the axes in your model at once, more so than another typical way that people do it will be they just take sort of outliers on each axis independently based on the loadings in the model, whereas this kind of incorporates all of our information at once. So it's a more sophisticated way than just looking at the outliers on each on the loadings on each individual axis, basically. There's there. Let's see if this one will run. Okay, cool. And this will generate a few different things. One of the things you want to look at here is there's a little bit of a frequency distribution of the p-values generated by this. So what you want to see is essentially what you've got here. You want a fairly flat distribution for the majority of your loci, and then an excess of really low p-values that represent your candidate loci here. Sometimes if you have, you might end up with more of a curved distribution, sort of a skewed distribution towards the left, then that's often an indication that population structure is still impacting your data, and perhaps you need to relook at how you are controlling for that. Not just that, but that's a common thing that can show up. And then the other thing you'll need to decide when you run that is what Q value threshold to use. So this is essentially a false discovery rate controlled p-value. And we can run code here that compares how many of those loci we consider as candidates based on how strict we want to be without Q value. And a Q value is essentially saying how many false positives am I prepared to accept in my data. So depending on your situation, you may be more or less strict with that. There's no sort of hard and fast rule there. And then the final code that I had was just, you might want to have an idea about, well, how are those candidate loci distributed across the genome? Are they all concentrated on 1 or 2 chromosomes? Or is it a more even spread that people tend to interpret as sort of consistent with polygenic selection? So if we run that code, yeah. Yep, I think the Manhattan plot one doesn't run now either. What happened? Oh, is it timing out maybe? It's yeah, but I'm in the tutorial session though. I was going to say, do you want to share your screen for that? Yeah. Okay. Sorry, people. I can't do that within the session.
Yeah, okay. Okay. So, this is just plot, and Chris nicely explained that it's always good to look at the 3D plot. First of all, it's cool, and second, if you look at the axis, now we have 1 and 2. That's what you normally see only in the 2D plot. In the 3D plot, you can see, more what's going on, and it's really nice to look around and see basically which of the environmental drivers are kind of linked to which of the loci, and you can see that better with linear 3D plot. Also, there was another thing which seems not to work in the tutorial, which is a bummer. So give me a sec. I had that here. No. Okay. There was also the Manhattan plot, which seemed to work here. Is that the Manhattan plot? Yeah. Give me a sec to run everything. Here it is. Yeah. Okay. For whatever reason, I have no clue, because it's installed, but somehow this library command seems to be missing. So please put that in there. Then I think this works. Yep. And this should have created a Manhattan plot in our under plots. Fun?
You just scroll back in the console.
Sorry, it's in there, is it? Yeah, sorry. There was it here. You want to say something to that? Or you?
Yeah, we'll just, as long as the code actually, well, I know it does run. Yeah, people can run that. Essentially, that's you need a map of where your SNPs appear on the chromosome. Typically a bed file or something like that. You can generate that from a VCF file, and then you can use the Q values or some other whatever in your particular approach you use to define the candidate loci on the y-axis, and then it will just plot. There's a dotted line there on the Q value for the example here, which I think was 0.01 or something like that. And you can sort of see that everything above the dotted line is the candidate loci. So you can see they're pretty evenly distributed across the genome in this case. It's really just some code there to get you going, and you can play with that and modify it as you need to for your data. So I think in the interest of getting some questions done, maybe we can just people can run through the rest. The only other code we had left to run was just using those candidate loci and then running another RDA at the individual level just on those loci, just so you can see how the individual samples fall out in that. Not sure whether. Yeah, maybe if I can share my screen again. Yeah.
So just for fun, I just run the code and you see it works. It's just I have to provide evidence to Chris that the code is not running. But as always, I think in the tutorial it seems to be too slow. So please, run the code maybe manually afterwards, and it creates beautiful plots. A bummer that you can't see them sometimes. Yeah. Okay. Maybe. If that's
we can just go to some questions now. Yeah.
Chris, do you hear me there?
Yeah, maybe we just decided we maybe go to the questions to wrap up the session.
I have already answered several of the questions. I was in the process of answering others, but Chris, can you look at the more RDA related questions and maybe you can answer those? There's still quite a few there.
Okay, I try to find them and put them up.
And I'm answering others as you go from the bottom up.
Can you see, Chris?
Oh yeah. Sorry, I was looking at the. Yeah, so I think in most cases, it's going to depend. I mean, it really depends on the analysis you're running. For running an RDA, for when you're interested in genotype environment association analysis, I think you're better to impute the data than to drop either the locus, because the overall effect of that is probably going to be minimal on the model compared with just having less data in it. So I think in most cases, figuring out the best way for your data, but trying to impute the missing data rather than just dropping it, I think is a better instance. Yes, it's very much the same in PCOA. So it's the orthogonal axis, so independent axis. The first one is the one that explains the most variation. The second most variation. So it's exactly the same as PCA or other ordination methods. That's a pretty good question. It was, I think when people first started doing these analyses, it was really common to see people would run, 2 or 3 or even more of these, and then
I've already answered that.
Okay.
Yep. Just in the interest of time, let's jump to stuff that hasn't been answered. Yeah, I'll just mark his answer. Yep. Sorry. Yeah, there's quite a few unanswered yet. No.
That really, I guess, in most cases at the level, both of the data we're using and the scale of the systems, you're usually looking at broad kind of environmental variation. And I think in most cases, you're going to find a more general signal across the genome than you will sort of, the more classic, I guess, islands of differentiation, or was framed as islands of speciation in the past. But you're less likely to find a signal concentrated in one chromosomal region at the level that most people are probably going to be doing these analysis. But it really depends on your species and your system.
I also want to add to that, if you have whole genomes, you might be picking structural variation as well. That might create a very different Manhattan plot. And if you're looking at in a region of major phenotypic effect, you also might, underlying important trait, you might be also seeing quite a strong area of divergence in the genome. It all depends on what you're comparing. For folks that are using similar approach in a kind of a GWAS context, you might have two phenotypes and you run your SNPs. You might be seeing not just a bunch of areas of small effect that give you a kind of a dense Manhattan plot, but you might be seeing some skyscrapers here and there with areas of low or no differentiation. It depends.
I think we have two more questions. Maybe let's go for them and then wrap up.
Not quite sure. I mean, essentially, it depends on the type of selection, I guess. The heterozygosity is potentially something that could provide a signal, particularly maybe if you're thinking about balancing selection. I don't know. I think you're better off using either the allele counts or allele frequencies in this case.
Just to add to that, I didn't have the chance. I had a plot exactly on this in one of the case studies. I think it's perhaps better to look at heterozygosity of your neutral and your candidate adaptive SNPs separately, as opposed to consider heterozygosity within the analysis. So you define your adaptive data set and you look at spatial patterns of heterozygosity, and contrast that with your neutral SNPs.
Second last question. Want another come in and then we stop?
Yes.
Sorry, guys.
Yes and yes. Simple answers there. Yes.
I guess depending on what you actually mean by filtering, but yeah, we're generally less stringent. I mean, there's, I guess, an argument for me. I don't filter for Hardy-Weinberg equilibrium before any of these tests because, I mean, you're looking for a signal of natural selection. That's one of the criteria that is this. The Hardy-Weinberg model is it breaks the model. So I guess yeah, I would generally be maybe perhaps less stringent in filtering this analysis, but it really depends on your data and minor allele frequencies. They're still important for depending on the depth of your sequencing samples. So you still need to filter in a fairly similar way, I think, to most other PopGen